Hi, Michelle Glass here. Welcome back to another one of our Chapter 23, Lecture 23 lecture topics. The topic for this video is gas transportation, and specifically we're looking at the movement of oxygen gas from the bloodstream to the tissues as needed. So let's get started. We're still working through our topic of external respiration, where we've talked already about the breathing, the inhale and exhale of breath, actually getting the oxygen into the body. Then we talked through the diffusion of gases, the determination of its movement from alveoli to blood to tissues, or from tissues to blood to alveoli. And then our topic that we have remaining is the actual transportation of both oxygen and carbon dioxide gas through the bloodstream. And again, this video is focusing specifically on oxygen gas transportation. Remember that our oxygen molecule is being transported in our red blood cells, specifically by the protein called hemoglobin. Remember your hemoglobin structure. You have 4 heme groups. At the center of each heme group, you have iron, and that's actually the thing that oxygen is attracted to. So that tells us that each hemoglobin molecule can bind and transport 4 oxygen molecules. When your hemoglobin is bound with oxygen, it's called oxyhemoglobin. And again, each hemoglobin molecule can bind to 4 oxygen molecules. Because our red blood cells are basically just stacks of hemoglobin, we can see them transporting as many as 1 billion oxygen molecules in a single red blood cell. So it's a very effective transport mechanism. When we talk about hemoglobin saturation, what we're talking about is how much how much oxygen is bound to that hemoglobin. So if we say that we have 100% saturation, then that means every single one of those irons at the center of each one of those hemes is bound with oxygen. So a hemoglobin molecule that's 100% saturated is a hemoglobin that's transporting 4 oxygen molecules. 50% saturation would mean instead of carrying 4 oxygen molecules, we're just carrying 2. So that's how we can interpret that information. Okay, as we look at our hemoglobin molecule, the question is, what are the factors that affected saturation? What's going to cause it to hold on to that oxygen or to release that oxygen? For me, as I first started imagining what was happening here, I imagined that the blood was moving through the body and then just delivering equal amounts of oxygen at each one of those capillary beds. That's not true. And of course it's not true, because how are you going to have enough oxygen to get all the way to the big toe if you're delivering oxygen all the way as you're traveling down? You're not going to get equal distribution, and your tissues are not equally active, so they don't have equal oxygen needs. So what we're going to be talking about here is what are those factors that determine what tissues get a lot of oxygen and which tissues don't. So here we're looking at our partial pressure of oxygen. That's, of course, what the PO2 stands for. When you have a tissue with a high partial pressure of oxygen, then that hemoglobin is going to hold on to that oxygen molecule. Because remember, based on our diffusion of gases, oxygen diffuses from a high partial pressure to a low partial pressure. So if there's a high partial pressure in your tissue, then there's not so much pull to get that oxygen away from the hemoglobin. The other thing here is this is helping that hemoglobin to bind up with that oxygen. So as the hemoglobin is moving through the capillary of the lungs and initially picking up that oxygen, the more oxygen that is binding to the hemoglobin, the more oxygen will bind to the hemoglobin. So as you get like this one oxygen molecule binding to a heme group, it changes the shape of your protein such that it's easier for a second molecule to bind, which makes it even easier for the third molecule to bind, which makes it even easier for the fourth molecule to bind. So we talk about this attraction between molecules as affinity. So as you get more oxygen, it's easier for that hemoglobin to actually hold on to and bind with that oxygen. Now, this also means that as your blood is moving through those inactive tissues, the inactive tissue is not using a lot of oxygen. So we're going to see that the partial pressure for oxygen is higher in the blood than it is in the tissues. And so the hemoglobin will hang on to the oxygen and it will not really deliver a whole lot to those inactive tissues or cells. This is really good because then as that blood moves along to tissues that are active, you have a smaller partial pressure of oxygen by those active tissues. So of course your oxygen is going to go from a high to a low partial pressure. So hemoglobin is releasing it to those active tissues. There are other factors affecting hemoglobin saturation besides partial pressure, which we've already discussed. We're going to see that blood pH has an effect and temperature has an effect. And this should not come as a surprise to you at all. Hemoglobin is a protein. Proteins are functioning based on their shape. Protein shape is affected by factors such as pH and temperature. So changes to these factors will change the shape of the hemoglobin, which in this case means its ability to hold on or release that oxygen. And we'll see also that metabolism of the red blood cells will have an effect on the hemoglobin saturation as well. So we'll be looking at these three factors in turn. Let's start with pH. This is a direct relationship, which simply means the arrows are going in the same direction. As you decrease your pH, you decrease your hemoglobin saturation. Decreasing your pH, remember, means a more acidic solution. Decreasing your hemoglobin saturation means you're going to be releasing your oxygen gas. Now, this relates also to active tissues. Tissues that are active are releasing carbon dioxide gas, which you already knew that. What you might not already know is that carbon dioxide gas combines with water to make carbonic acid. Carbonic acid is an acid, so it is going to lower the pH of the surrounding fluid. So again, here we're seeing a metabolically active tissue changing the environmental conditions, making it more acidic. When this happens, the hemoglobin is going to release the oxygen. So this increase in metabolism, this release of carbon dioxide gas, making the solution more acidic or lowering the pH, is going to be a trigger for red blood cells, hemoglobin, to release oxygen. Now this is described as the Bohr effect, this effect that pH has on hemoglobin's release of oxygen. Again, not only are we seeing more oxygen released to active tissues because of the Bohr effect, but we also saw it earlier because of the difference in partial pressure. So here's two ways that active tissues are pulling more oxygen basically out of the bloodstream compared to our tissues at rest. Okay, now let's look at temperature. Increasing your temperature decreases your hemoglobin saturation. So remember, this is going to be an inverse relationship where our arrows are going in the opposite direction. Again, this relates to active tissues. Tissues that are active are generating heat. If you're generating heat, then as the blood is moving through that tissue, it's going to trigger that hemoglobin to release the oxygen. So now we've seen the partial pressure of oxygen pulling more oxygen out of the blood to active tissues. We've seen a lower pH in active tissues, pulling more oxygen out of the blood. And then now we're seeing temperature as another way that we can get those active tissues to really pull that oxygen out of the bloodstream. Oh, I hit the wrong button. Sorry about that. Okay, so that's what we're looking at here. Those active tissues are using oxygen, they are releasing carbon dioxide gas, and they're generating heat. These are all factors that are going to decrease your hemoglobin saturation. So as the blood supply is moving through active tissues, more oxygen is being released to those tissues because they need more oxygen. Those tissues at rest have a higher pH, they have a higher partial pressure of oxygen, and they have cooler temperature. And so, yes, of course, some oxygen is being delivered there, but not quite as much. So this kind of explains how your blood can move all the way through your body, delivering different amounts of oxygen to those active tissues versus those resting tissues, so that there is enough oxygen to kind of get around, so no tissues are getting cheated. Okay, so we've talked about how blood pH and temperature affects hemoglobin saturation. Our final topic is the metabolism of those red blood cells. Now, one thing to pay attention to, red blood cells are lacking most organelles. They don't have mitochondria. They can't do cellular respiration. They're limited to that process called glycolysis. As a result of the glycolysis, your red blood cells are producing a compound called BPG for short. It's 2,3-biphosphoglycerate is the name of the molecule that's there in your textbook. But for us, we can just think about it as BPG. So it's a lot easier to write down and to say. Okay, this is another inverse relationship. The more BPG being produced by that red blood cell, the lower the hemoglobin saturation. So that means it's releasing that oxygen for tissues. So metabolically active red blood cells roaming through the body are making normally a lot of BPG, and so this is letting them release that oxygen as needed as these cells are moving through the tissues. In fact, we'll see that there are situations where the red blood cell will actually increase the amount of BPG in order to keep that oxygen flowing out to tissues. It's kind of a homeostatic regulation system. As red blood cells get older, remember we've already talked about them not being as flexible, not moving through the capillaries the way that they should, being removed by those macrophages before they burst open in the bloodstream. But here's another factor. As those red blood cells get older, they are less metabolically active, which means they're not producing that BPG. As BPG levels decline, these hemoglobin molecules are holding on to the oxygen instead of letting go. This is another reason why donated blood has a shelf life. Ultimately, those red blood cells start to age on the shelf, and they hang on to that oxygen. They don't let it go. So there would be like no point in giving old blood to a person, because you're trying to give them these good quality oxygen transporters. And as those red blood cells get old, sure, they're moving through the bloodstream, and sure, they're carrying oxygen, but they are not letting it go. And so clearly that's not going to be an effective way to get oxygen out to your tissues. All right, and that's it for now.